So in this video, I am continuing where I left off in the previous video. Now, the previous presentation involved chromosomes and the cell cycle. So now we will be going into actual mitosis. So I want to remind you that the function of mitosis is cellular reproduction or division, but that cellular reproduction is used for three different things: asexual reproduction, growth and development, and tissue repair. Again, I strongly urge you to watch this video. I think it is very helpful. Okay. Now, there are 4 stages or phases to mitosis. So the first thing that I would like to point out here is this phase that we call prometaphase. So we will not be going into prometaphase. Some textbooks include prometaphase, others do not. Now, there are 4 phases to mitosis: prophase, metaphase, anaphase, telophase or telophase. And then there's one additional process called cytokinesis. Okay. Let's look at here. Okay, so this is prophase. Now I'm going to list the items that you need to know about prophase and draw a picture for you on the whiteboard. So I know there's a picture here, but I'm going to illustrate it in a slightly different way that I think will help. Okay. So this is prophase, the first stage in mitosis. Okay, so for prophase, there are 3 main things that you need to remember, and these occur during this stage we call prophase. So the first one is that the nuclear membrane, and you will also see it called a nuclear envelope, breaks down. Okay, so throughout mitosis, there is no nucleus. The next part is that the chromosomes, now remember, each chromosome consists of 2 sister chromatids. So the chromosomes supercoil or condense. They become really tightly wound around those histone proteins. And this is where they take on that traditional X formation that you are used to looking at. Now, finally, a formation that we call the mitotic spindle forms. Now, the mitotic spindle is where microtubules grow for, we call it division of the genome, for separating those sister chromatids. Remember, your genome is all of your DNA. So division of the genome is taking one set of DNA and separating it from the other set. So I'm going to draw a picture of this. Now, with humans, I told you before that we have 23 pairs of chromosomes. I'm going to draw 2 different pairs in my image here. Okay, so those would be sister chromatids from mom. The long ones would be from chromosome pair number 1. The short ones would be from chromosome pair number 2. And they're not in any particular formation for prophase, but draw them like this. So the long red and blue sister chromatids represent chromosome pair number 1. Now remember from the last video, I showed you chromosome pair number 1 and it coded for eye color, okay? Now, the short red and blue sister chromatids represent chromosome pair number 2. Now let's just say that chromosome pair number 2 contain the genes or gene for height. I'm going to draw some genes on here. So here we would have that gene for eye color. Here we would have the gene for eye color. So the 2 long red lines here represent sister chromatids from mom. The 2 long blue lines represent sister chromatids from dad. So here we would have brown eyes, right? And then this would be brown eyes. Now the long blues are homologous to the long reds. So here we have blue eyes and blue eyes. Okay, so the long reds are homologous to the long blues. They are not identical, but they code for the same inherited traits, such as eye color. Now let's go down to the lower pair. Made these so small, it's going to be hard to draw a gene on them. Okay, well, I'm going to not draw that gene. Okay, ignore that little black line there. But for these small chromosomes here, let's say that the gene on it codes for height. So let's say that the short blue ones code for tall and the short red ones code for short. Okay, both of those code for the same gene, height, but they have different versions or alleles. So the long blues are homologous to the long reds. The short blues are homologous to the short reds. So long blue sister chromatids, homologous to long red sister chromatids. And the same holds true for the short ones. So the long ones represent what would be chromosome pair number 1. The short ones represent what would be chromosome pair number 2. Okay, so going back to the picture of prophase here, what you are looking at are sister chromatids that are homologous to another set of sister chromatids, right? They're just hanging out in the cell there. The nuclear membrane is breaking down. This mitotic spindle forms right here That's the mitotic spindle, and it is made of microtubules. Remember, those are part of the cytoskeleton. And then finally, the sister chromatids become supercoiled or condensed. So now we are going to move on to metaphase. Now, I always think of metaphase middle. Okay, so what I mean by middle of the cell is that the sister chromatids line up in the middle of the cell. So, oops, let's go to the whiteboard. Okay. All right, so we are looking at metaphase. So with metaphase, the microtubules from the mitotic spindle attach to each sister chromatid. Okay, now the other thing that occurs is these sister chromatids line up in the middle of the cell at what we call the imaginary metaphase plate. So okay, now I'm going to go back to the slide. I'm not going to draw a cell because I have a really good picture of it on the PowerPoint. Okay, now notice that the sister chromatids are lined up in the middle of the cell. Now it says spindle fibers. Those are the microtubules. Now notice that spindle fibers from one side, where you see like those little cylinder structures, comes down and attaches to one of the sister chromatids in the orange pair. A microtubule from the other pole of the cell grows down and attaches to the other sister chromatid. So the metaphase middle. Sister chromatids line up in the middle of the cell. Now, when the cell eventually goes through separation, it separates along that metaphase plate. So one set of sister chromatids will be pulled to the left side, one to the right. Those are what we call opposite poles of the cell. Okay, now anaphase, that is the first or third phase of mitosis. So for anaphase, what you need to remember is that sister chromatids are pulled apart towards opposite poles of the cell. Now I actually am good. Well, I don't think I need to draw this on the whiteboard. So let me write this down here. Now it says daughter chromosomes on the picture. Don't worry about that terminology. It's not wrong, but it's a little more detailed than we need to go in terms of like terminology, at least. Okay, so during anaphase, sister chromatids are pulled apart. I'm going to add this by shortening microtubules to opposite poles of the cell. So what's happening here? Remember, the microtubules came out of those little cylinder structures at each side of the cell, right? The microtubules attached to each sister chromatid in the pair, Now, those microtubules are actually going to start breaking down and pulling back towards those cylinders. And that pulls those sister chromatids apart at the centromere. Okay, so this is in preparation for division. So you can see here in the picture, those sister chromatids are separated and they're being pulled back. Now, once they are separated, they are called chromosomes again. And I know that's a little bit confusing. So when sister chromatids are attached at the centromere, they're considered one chromosome. Once they're pulled apart, they're now two chromosomes. Now, this is the last official stage of mitosis. And I say that because there is still a process called cytokinesis next, which is not actually considered part of mitosis, but it's really kind of part of the whole process. So during telophase, I'm going to write this down for you, but the nuclear membrane or envelope reforms, right? So now you have two nuclei. The cell starts to pinch in half and forms what we call the cleavage furrow. And the cell is pinching in half because a microfilament that's part of the cytoskeleton is causing it to pinch in half, kind of like a drawstring. And the chromosomes begin to uncoil. They are less dense. I'm going to write this down for you. So the nuclear membrane reforms. So now there are technically two nuclei in one cell. Now the nuclear membrane reforms around those two sets of DNA, right? One forms around one set, one forms around the other set. Chromosomes, I don't know if this takes a hyphen, decondense. And microfilament begins to pinch the cell in half, forming the cleavage furrow. Okay. So far you have learned about prophase, metaphase, anaphase, telophase, but we still do not have two cells yet. That occurs during cytokinesis. So here is cytokinesis. Now, technically, this is called division of the cytoplasm. Now, this is when the actual cell is fully split in half. Right here, I'm going to write division of the cytoplasm. Okay. It doesn't show the cell fully pinched in half in this picture, but that's what happens by the end of cytokinesis. Now, each resulting cell, we call those daughter cells, have one nucleus each with the exact same DNA that the original parent cell had. So let's look at this. So let me give you a couple big picture things to remember here. Okay, so this is like general information about mitosis that you need to remember. If I can get my text bar to work, there we go. So in mitosis, you go from a parent cell, you have mitosis, and you end up with two identical daughter cells. They're identical to each other and they are the same as the original parent cell. Okay. So one parent cell, two genetically identical daughter cells. Well, I'm going to draw a picture here to try to show you how this works. So here is my original parent cell. Make that black. Okay, that's my original parent cell. I'm actually going to draw my cells and then I will fill in stuff. Now, these cells are actually all the same size. I'm just running out of room. So here we have this. I'm going to explain this after I fill in my chromosomes here. Okay, so take a look at the cell to the far left. This is my parent cell, like the original cell that is going to divide. Okay, so this is my original parent cell. It has homologous chromosomes, no sister chromatids yet. Now, what I drew here would be like chromosome pair number 1. Now, remember, in a human, we have 23 pairs. Now, we will have S phase of the cell cycle, which hopefully you remember is DNA replication, right? So this is not mitosis. This is part of interphase, G1S, G2. Now during S phase, the DNA replicates. If you're confused on that, go back to the presentation on DNA replication. So after S phase, there are sister chromatids for each pair, okay? Now the cell will go through mitosis. So when I say mitosis, I mean prophase, metaphase, anaphase, telophase, cytokinesis. Now you end up with 2 daughter cells. Okay. So we started with parent cell, went through S phase, doubled the amount of DNA, went through mitosis, and that restored the original amount of DNA. Now, I want to clarify something here. I said that these are identical, genetically identical daughter cells. Well, all cells have the exact same DNA, right? Actually, I should say all cells have the exact same genes in them, right? But the way that these genes are controlled results in different types of cells, right? Your skin cell has the same DNA as, say, a cell in your heart. But the way that the genes in the DNA are turned on or turned off at different points makes the cell become a skin cell versus a heart cell. Now, we will go into this more during the genetics lecture, but I want you to realize that your cells all have the same DNA. It's just the things that control the genes that are different in your different cells. And when I say turn on or off, what I mean is transcription and translation, right? A gene is not always being transcribed. It is not always being used to make a protein. Sometimes it's just not active and no protein is being made from that gene. So the example that I like to illustrate this for those of you who have kids or even seen babies, babies are born with kind of a grayish color eye. And right around, I think it's like 6 months, it might be a little earlier than that, their eyes change color, right? Like some kids get brown eyes, some kids get blue eyes. That is because the gene for eye pigmentation does not become active until that age. At the age at which your child's eye color changes, that is when the pigmentation gene turns on and starts making pigment proteins. Prior to that, it's turned off. So that is an example of how genes turn on and off. Okay, so that is it for this lecture. Next, we will be covering meiosis, which is also cellular reproduction, but meiosis is used in sexual reproduction.